Hi everyone, so we'll get started with session 2, fibrosis and wound healing. And so today the moderators are Dr. Sarah Cuevas-Ocaña and Dr. Michael Blatchley from the University of Nottingham and University of Colorado.
Hello everybody. We're very excited to introduce you to session 2 of this great symposium. I'm Sarah Cuevas-Ocaña from University of Nottingham, UK. And I'm going to pass over to my co-moderator, Michael, who's going to introduce the two first speakers, and I will introduce the second two. Hope you enjoy this session.
Thanks, Sarah. Hi, everybody. My name is Michael Blatchley. I'm a postdoc in the ANSEF lab at the University of Colorado in Boulder. And as Sarah said, I'll be introducing the first two speakers. The first is Natalie Khalil, who will be talking about her investigation of oxygen-dependent cell-cell communication in human cardiac fibroblasts with a microfluidic device. So you can go ahead and share your screen, Natalie, and we're looking forward to your talk.
Thank you. Thank you for the introduction and for the opportunity to present today. My name is Natalie, and I'm beginning my second year in Professor Megan McCain's Laboratory for Living Systems Engineering at the University of Southern California. And I'll be discussing oxygen-dependent cell-cell communication in cardiac fibroblasts. So to begin with, heart disease is currently the leading cause of death worldwide and is responsible for over 17 million deaths annually. It's primarily caused by plaque buildup in the coronary arteries, which deprives the myocardium of vital oxygenated blood. Complete occlusion of the coronary arteries, as pictured here, leads to localized hypoxia in downstream tissue, and this is known as a myocardial infarction or heart attack. So immediately in response to this hypoxic injury, approximately 1 billion cardiac myocytes will die within hours. And since the mammalian myocardium has limited capacity for regeneration, the subsequent repair process is dependent on the formation of a scar. And this occurs through 3 phases. In the first few days, in the inflammatory phase, neutrophils and macrophages will clear dead cell debris and matrix fragments. Over the next few weeks, during the proliferative phase, cardiac fibroblasts will expand and then activate into a myofibroblast phenotype. And myofibroblasts will play an important role in depositing the matrix proteins, which form the scar in this final healing phase over the next weeks to months. However, there are many different cellular and molecular events that may take place during this process, such as excessive fibroblast proliferation or an imbalance in the synthesis and degradation of matrix proteins that all may lead to excessive myofibroblast persistence or survival in the myocardium. And while myofibroblasts are essential to maintain tissue integrity and prevent myocardial rupture after infarction, excessive or pathological fibrotic remodeling by myofibroblasts is ultimately associated with arrhythmias or heart failure. So finally, the border zone after a myocardial infarction is defined as a small region of tissue situated between the hypoxic infarct zone and normoxic neighboring viable tissue, and is characterized by a gradient of oxygen tensions. Recent publications have investigated crosstalk between hypoxic and normoxic cardiac cells that may regulate things like cardiac myocyte viability or cardiac fibroblast migration. But conventional in vitro models have typically been limited to studying a single global oxygen level through the use of a hypoxia chamber or an incubator with gas oxygen regulation, and also have typically been limited to paracrine signaling factors through the transfer of conditioned medium from hypoxic to normoxic cells or vice versa. So therefore, existing in vitro platforms do not recapitulate real-time signaling that's observed over physiological oxygen tensions in the heart after an infarction. So we aim to engineer a border zone on a chip for oxygen control to cardiac cells in order to investigate oxygen-dependent cell-cell signaling after an infarction. And so in aims 1 and 2, we'll ultimately characterize the cardiac fibroblast response to uniform and then gradient oxygen as in the border zone. And then finally in AIM-3, we'll look at crosstalk between hypoxic cardiac myocytes and normoxic fibroblasts and vice versa. So first, to characterize the response to uniform hypoxia, we will expose cardiac fibroblasts to global 0% oxygen for various time points. And then to assess activation into a myofibroblast phenotype, we'll stain for alpha smooth muscle actin, which is an actin isoform that is a marker of myofibroblasts that enables them to generate force for wound contracture. And then to assess the response to hypoxia, we'll stain for hypoxia-inducible factors, which are a family of transcription factors that in normoxia are hydroxylated by an enzyme called prolyl hydroxylase, and this marks them for degradation. But in hypoxia, since they cannot be marked for degradation, they'll dimerize with their beta subunits, enter the nucleus, and act as transcription factors to upregulate a variety of genes associated with survival in low oxygen. So in addition to looking at activation into myofibroblasts and HIF activity, we also want to look at other phenotypes relevant to infarct healing, including fibroblast proliferation, migration capacity, and cell viability, and also confirm some of these at the mRNA level. So far I've seeded human cardiac fibroblasts and exposed them to 0% oxygen. And you can see that by 2 and 4 hours of global hypoxia, HIF1 significantly accumulates in the nuclei of primary human cardiac fibroblasts, where it may be acting as a transcription factor. And this is quantified in the figure on the right. Interestingly, with HIF2, we already see some accumulation in the nuclei at baseline, and it doesn't seem to be affected by hypoxia treatment. And this may be for, I guess, 2 reasons. Firstly, HIF2 has been reported to be involved in the chronic response to hypoxia, so perhaps its activity cannot be captured in under 24 hours. And then secondly, HIF2 has been reported to be less efficiently hydroxylated and therefore less efficiently degraded in this kind of normoxic control condition prior to hypoxia treatment, which may explain why it's accumulating in the nucleus at this time point. And then lastly, we've tried to look at activation into myofibroblasts by staining for alpha smooth muscle actin, and we did see some upregulation by 24 hours, in which the data is not really shown here. But as you can see in the normoxic control condition, there's already some basal levels of alpha smooth muscle actin expression. And this may be because I was culturing the cells in 10% serum, and serum contains cytokines such as transforming growth factor beta that might trigger fibroblast activation. So I'm currently repeating these experiments in uniform oxygen with serum arrested cells. But once that's completed, we could engineer an oxygen gradient and expose cells to gradient oxygen to understand how oxygen regulates fibroblast activation. So to do this, we can engineer a microfluidic device with 2 channels for gas flow. In one channel, we can flow hypoxic gas or 0% oxygen, and in the other channel, we can flow physoxia of the myocardium, which is around 10% oxygen. And these channels are made using photolithography and soft lithography techniques. Channels are sealed with a PDMS membrane that is 100 microns thick, and PDMS is gas permeable, so gas can diffuse across the membrane and reach cells that are cultured on top of it. And then in addition to this, we can modify the device with PDMS well designs that can control different modes of cellular communication. So in the far left, cells are seated in this kind of one giant well, so that regardless of oxygen tension, they're in direct cell contact with one another and can undergo paracrine signaling through media. On the far right, these low barriers inhibit direct cell contact between the wells while still enabling paracrine signaling through media. And in the central device here, we're inhibiting both forms of communication. So this will allow us to investigate how paracrine signaling and direct cell contact over physiological oxygen tensions regulates fibrotic mechanisms. So next, to characterize surface oxygen levels to validate the gradient, we can engineer an oxygen we can use an optical oxygen sensor called PTOEPK, which is very fluorescent in the absence of oxygen and becomes quenched with increasing oxygen concentration. So we'll deposit PTOEPK onto the device, take images of its fluorescence along the gradient, and then use these fluorescence values to solve the Stern-Volmer equation, which relates fluorescence in the absence or over varying oxygen concentrations to concentration of oxygen along the gradient. So this is very preliminary data for one device, and it is lacking calibration images, so the numerical values are not relevant here. But you can at least see qualitatively that along the hypoxic channel, PTOEPK is very fluorescent, and it becomes quenched as you approach normoxia, and this is seen with the pixel intensity below. And as pixel intensity is decreasing along the gradient, we can compute that oxygen is increasing as you approach the normoxic channel. So this would need to be repeated to demonstrate a linear increase from 0 to 10% oxygen. So next, we could expose cardiac fibroblasts to the 3 devices for 4 hours based on uniform oxygen experiments to observe HIF activity, or longer time points to observe activation into myofibroblasts with homogeneous normoxia and hypoxia devices as controls. So for one device in which the gradient is not yet validated, we do see differential HIF activity along the gradient with fibroblasts along the hypoxic channel having HIF accumulation in the nuclei. And this is supported by previous work with endothelial cells that were also exposed to a linear oxygen gradient, where oxygen is shown in green here. And near the hypoxic end of this device, at around 2.5% oxygen or lower, this endothelial cell line had significant HIF-1 upregulation in the nuclei. Something that would be interesting to observe is this concept of hyproxia or hypoxia by proximity. And this was seen with HIF-2 in the same endothelial cell line. So again, along the hypoxic end of this gradient, we do see this endothelial cell line upregulating HIF-2 approximately 1.6-fold higher than a homogeneous normoxia control device. But even as you approach the normoxic end of this gradient, normoxic endothelial cells have approximately 1.5-fold higher HIF-2 accumulation in the nuclei than the normoxic control device. And so this indicates that there's some communication occurring between hypoxic and normoxic endothelial cells that is stabilizing HIF-2 even in normoxic cells on this gradient. So if this is occurring in cardiac fibroblasts, this would potentially play a role in activating even normoxic fibroblasts. And so lastly, in the future, we want to also look at crosstalk between hypoxic myocytes and normoxic fibroblasts and vice versa. And so to co-culture hypoxic and normoxic cells, we'll engineer a very similar microfluidic device where we've decreased the diffusion gap between the hypoxic and normoxic channels to create a very steep gradient in between the 2 regions. So this essentially creates a stepwise increase from hypoxia to normoxia in this case. And another modification that we'd like to incorporate into this aim is muscular thin films, which can allow us to directly measure cardiac myocyte contractile stress in the 3 different modes of communication with fibroblasts and also in hypoxia versus normoxia. So these experiments will let us know whether expected phenotypes of hypoxic fibroblasts, which we kind of will determine in aim 1, are validated or may be altered based on communication with myocytes. So in the future, or I guess kind of more broadly, this work could potentially identify molecular targets for anti-fibrotic therapies. The system could be used as a drug screening platform to see how cells exposed to different oxygen tensions respond to the same anti-fibrotic drug to kind of screen for more effective drug treatments. Seeded with patient-derived cardiac cells, this could provide insight into a patient's genetic contributions to their disease or drug response. The system could incorporate more physicochemical cues relevant to the infarct microenvironment, such as stiffness. And then lastly, the microfluidic device could be used to study other hypoxic tissues, for example, to look at crosstalk in the tumor. So I would like to thank Professor Megan McCain and the rest of the Laboratory for Living Systems Engineering, and particularly Dr. Megan Rexis Hall, who's a postdoc in our lab, who's taught me a lot about oxygen control. And so now I'll take any questions. Thank you.
Thanks a lot, Natalie. Really great talk. Sounds like you have a lot of really interesting experiments ongoing and upcoming. So I think we have time for maybe one quick question, and then maybe you can answer a few of the other ones after. So one question is about growth factor absorption in PDMS, and have you sort of accounted for this or controlled it in any way, especially with looking at crosstalk between cells?
That's a very interesting question. We have not considered that potentially secreted growth factors or other forms of paracrine signaling could be absorbed into the PDMS device. But that's great feedback, and that is something that I'd like to look into doing. Thank you.
Great, thanks. I think in the interest of time, we'll move to our next speaker, but thanks again, Natalie. So our next speaker is Snehal Kadam, and she'll be talking about a human-relevant in vitro bioengineered wound device to replace animal preclinical testing for therapeutics. So you can go ahead and share your screen.
Okay, can you guys see my screen?
Yep, looks great, thanks.
Okay, all right. Thank you so much for the introduction, Michael. I'm going to start off by just saying that this project that I'm going to be talking about today is a human-relevant in vitro wound device that we've been developing in the lab, and it is an ongoing project. So I'm going to be just presenting a little bit of preliminary data that we have, but I'd love to hear feedback and suggestions from everybody. So I'm going to start off by asking everyone to remember the last time you were wounded. And this could just be a small paper cut you got, or it could just be a major wound from a fall that you took. And while this can be a pretty unpleasant memory for most of us, our wounds would have healed normally. And wounds heal normally through various stages and can take up to some days or even weeks, depending on the size of the wound as well as the extent of the damage. However, there are some wounds that don't heal and become chronic. So chronic wounds are a major healthcare burden, and they're associated with a reduced quality of life, and they can be aggravated by underlying conditions such as diabetes. Billions of dollars are spent in healthcare costs worldwide, and they're really a global health problem that we are facing. One of the major reasons that chronic wounds become chronic is due to bacterial infections. These bacterial infections are most often present as polymicrobial biofilms and are the single most common cause for impaired wound healing. The presence of biofilms in wounds, given their increased antibiotic tolerance, also calls for the need for more effective therapeutic approaches in treating these chronic wounds. So as you can see, chronic wounds are a major problem, and being able to study them in the lab is really important to address these infectious agents and how we can deal with them. So the wound infection microenvironment consists of numerous components that interact with each other. This includes the biofilms, of course, that are infecting, the damaged skin host cells such as fibroblasts, keratinocytes, the recruited immune cells, all the signaling molecules that are secreted by immune cells as well as by biofilms in terms of their virulence factors, the underlying extracellular matrix of our skin, which is also usually damaged in wounds, as well as various biophysical forces that are present, such as shear stress, stretching, which have all been shown to be important for wound healing. So all in all, the wound infection microenvironment is a complex system, and it becomes very difficult to replicate this in vitro. So most current models use animal studies, and these include both ex vivo as well as in vivo models. But as you can imagine, these of course come with their own limitations, and they are not very human relevant when it comes to skin physiology. So there's really this need to bridge the gap between the in vitro and in vivo models that we currently have. And in order to do this, you would not only want a model that can recapitulate various microenvironmental factors, but also allow us to be selective and precise in terms of controlling how and when these factors are present in our models so that we can understand their role in wound infections much better. We would obviously want something that is more human relevant and that will allow us to reduce the scientific as well as ethical limitations that come with animal model usage. And most importantly, we would want a system that will allow us to potentially test various therapeutics in order to treat these biofilms that cause chronic wound infections. So in order to try and answer this question, our lab has been working on developing something we called a macrofluidic wound device. And we call this macrofluidic mainly because of its size. As you can see from the picture of the prototype, it's almost the size of a palm, but the size of the wound bed or wound area within this device has been designed based on the average wound size that has been reported in literature for a wide variety of wounds. And the main reason that we decided to come up with this macrofluidic wound device is because though microfluidic devices are great, we rarely get wounds that are micro in size. Almost every time you're injured, it's going to be something that's, when you fall, say, from a bike, it's almost, you injure something that's almost the size of your entire knee. So we would want something that's slightly bigger and that is able to replicate some of these physical and biophysical forces that act in the wound infection microenvironment. So currently we are using PLA just to prototype and optimize our design, but eventually we will be using polycaprolactone, which is a biocompatible polymer and has been shown to support the growth of fibroblasts. As you can see from the design, once printed, the top and the bottom of the device are actually open, but they have areas that we can fit cover slips in, and we can use either a sealant or a gasket or even a 3D printed frame to make sure that this junction is airtight. It has an inlet and outlet which connect to tubings that allow us to establish flow in our device. And just the size of this device and the fact that it has a wound area that has a transparent bottom allows us to directly use this for imaging as well. Now, as I mentioned, it has an inlet and outlet that allows us to establish flow. And in order to control this flow, we also developed an in-house cost-effective peristaltic pump. And this pump is completely battery operated and it's modular and it can be electronically controlled to set flow rates as well as time. It has silicone tubing that connects to the inflow of the macrofluidic device as well as to your media reservoir. And the entire setup of the device as well as the pump together is almost the size of a shoebox and it can fit into a CO2 incubator. With the current design of our peristaltic pump, we are able to achieve flow rates with a minimum of 10 microliters per minute and to a maximum of 2 ml per minute. And when we take into account the size of our wound device as well as these flow rates, theoretically, we can achieve shear stress values of a minimum of 0.03 dyne per centimeter square and a maximum of 0.65 dyne per centimeter square. As you can see, this is a pretty large range, and previous studies have reported values that fall within this range that are known to promote healing in other in vitro models. So the 3D printing of our entire microfluidic device takes about 4 hours. And though currently we're using PLA, once we move to polycaprolactone, which has a low melting point, this cannot be autoclaved. So we right now just clean this with ethanol washing as well as UV sterilization. The silicone tubings and glass cover slips that are part of both the device and the tubings that connect to the pump are all autoclavable, so they can be sterilized. And this whole circuit of the pump, the device, the connected tubings, as well as the media reservoir are aimed to form a closed circuit and maintain sterility. And this is mainly done because we assemble this whole thing inside a biosafety cabinet. So now we've designed a device that we think is more appropriate in terms of its size and its various aspects of flow that allow us to be able to relate it to wounds better. The next important step is to incorporate the various microenvironmental factors that are present in chronic wounds into this device. And so we decided to start off with the most basic necessity, which was the skin host cells. And these include fibroblasts as well as keratinocytes. So one of the major issues with co-culturing both of these cell lines is their differential media requirements. So fibroblasts require around 10% serum for them to grow well and proliferate, and keratinocytes generally prefer serum-free media. So current methods that do co-culture these usually have protocols where there are sequential changes of media, where initially fibroblasts are grown in 10% serum, and then once the keratinocytes are introduced, the media switched out to a serum-free media. As well as there are more complex protocols where different growth factors are added at different time points in the protocol. There are feeder systems, there are irradiated cell lines. And there are very few approaches that actually allow both cell lines to be seeded simultaneously. And while a healthy skin would have fibroblasts and keratocytes layered, a wound bed is not really healthy skin. So there are definitely going to be pockets within the wound bed because cells are migrating, where both of these cells are in close contact with each other. And we think a co-seeding approach is much more relevant in those pockets when we try to replicate them in vitro. So in order to grow these two cell lines together, we came up with two media formulations which have reduced serum around 1 or 2%. And we were able to show that these reduced serum formulations can support both a layering approach where fibroblasts are seeded and keratinocytes are seeded on top of the confluent layer of fibroblasts, versus a co-seeding approach where both are seeded simultaneously. In both of these approaches, both the cell lines were able to grow and proliferate and form characteristic features that have been seen in previous studies in the wound bed. Both our reduced serum formulations use very easy to procure media, and all of them are an all-in-one ready to use media. So there isn't a lot of addition of growth factors that multiple steps involved. And we were able to do this in tissue culture flasks as well as in 6 well plates. And when we do this in well plates, we can also do a wound scratch assay. So once we do wound scratch assays, we can observe wound closure in vitro. And that's exactly what we did observe, which indicated that our reduced serum formulations, even though they have only 1 or 2% serum, were able to support the growth, proliferation, as well as migration of both of these cell lines in vitro. Now, this is about as far as we've got before the lockdown here. And our next step is to be able to replicate this within our wound device. So this is in terms of being able to grow our skin cells within our device. But our proposed wound infection on chip model basically has various components, some of which we've already worked on, such as the wound device and the peristaltic pump, which I talked about today, as well as the co-culture. However, there are other parts which I will not be talking about today, but which also include growing biofilms in vitro. And as you can imagine, biofilms that grow in wounds don't really grow in LV. So we don't think LV is a really good media to use when we're trying to make our platform more human relevant. So in the lab, we've been trying to grow biofilms in a wound milieu that we've made. And we do see differences in terms of biofilm characteristics as compared to standard LV media. So our next step is to be able to incorporate these biofilms into our wound device. And doing this, we hopefully will be able to achieve host microbe co-culture, which we've done previously with planktonic cultures and shown that these bacteria do attach onto our fibroblasts and keratinocytes. So with this proposed wound infection on chip, we hope to create a platform that is cost effective and reproducible, but also allows more precise control over the various microenvironmental factors that are present in the chronic wound infection. And with being able to co-culture our wound bed, as well as adding microbial biofilms on top, we hope to be able to create an actual wound within our wound infection on chip. And hopefully in the future, this will enable us to test various novel therapeutics as well as in combination with therapies that are already used to treat these biofilms and help us make some progress towards treatment of chronic wound infection. And with that, I would like to thank my PI, Dr. Karishma Kaushik, who has been amazingly supportive through this project. And Jay, who's an engineer, he's been helping us with the 3D printing aspects of the wound device. Aniket is a student at IBB in Pune, and he has been helping us with the peristaltic pump. And Vandana is my colleague from the lab, and she's been helping with the co-culture experiments that we've been doing. I would also like to thank the university for all of their support, as well as Venture Center, who've been allowing us to use their 3D printing facilities. And finally, DBT Ramalingaswamy Reentry Fellowship for all the funding for this project. So thank you, and I will take any questions that you have.
Great, thanks for another great talk. Again, we have time for maybe one or two quick questions. First, have you looked at any sort of serum replacement formulas as another kind of fix for these co-cultures? So rather than just low serum, these serum replacements?
So we've not looked at serum replacement specifically, but when we were trying a reduced serum approach, obviously the first idea was to have a zero serum approach. And that doesn't work out well because the fibroblasts do not do very well in no serum, when there's no serum at all, despite having media that has other components in it. So there is some amount of serum that is required because these are primary cell lines. So we've been trying to play around with that a lot, but we've seen that this 1 to 2% is what sort of fits the optimum between none and too much.
Okay. And then another question is, how close is this chip to human skin injury, especially with the use of cell lines? And then maybe kind of along with that, have you thought about incorporating some of the other components of the wound microenvironment, like you described on one of the first slides, such as oxygen or other cell types?
Yes. So right now, like I said, we've just started this project. So we've been starting with cell lines, and we hope that we'll be able to create wounds with them. But obviously, we want to be able to replace this eventually with, for example, cell lines that are more relevant, like the HECAT is actually an immortalized cell line. So we would want to replace that with primary keratinocytes. And because we can establish flow in our device, we want to start off by flowing in different components that can make it more relevant to different wounds. So for example, if we want to study a diabetic wound, we should be able to flow in a milieu that is rich in glucose. And we want to be able to adapt. Basically, it allows us to adapt the platform to the kind of wound that we are studying.
Okay, thanks. There's a couple more questions in the Q&A that if you have time, if you could answer, that would be great. But with that, I'll pass it on to Sarah for the last two speakers of this session.
Thank you.
So to continue with this great session, it's my pleasure to introduce Dr. Evan Walby, hope I pronounced it right, and he's going to talk about this self-assembling 3D aspirate cultures of human neonatal keratinocyte with enhanced regenerative properties. Evan, when you're ready.
Thank you for that kind introduction. I'll share my screen with you all. Okay, let me know if it's working.
Yes.
Perfect. So yeah, so the work I'll be talking about today is work I started as a pre-doctoral student at the University of South Carolina School of Medicine, and moved as a postdoc and expanded that work. And I'm actually thinking about it, taking it also on my faculty, from my own laboratory and the faculty. And so most of what I'll be talking about is a very simple system to study wound regeneration, specifically in the context of the skin. So about 6.5 million patients annually are affected by wound injury. The cost for our healthcare system is about $25 billion as of 2014, has obviously increased since. And so there's a critical need for high throughput ex vivo models to actually be able to study this disease. One of the limitations that we've had so far is most of the models, with the exception of in vivo models, most of the ex vivo or in vitro models rely on studying the step of wound healing in individual phases. But we know that process occurs in a sequential manner. And so we wanted to find a method to capture that sequential flow put, if you will, in culture. And so typically what you would have after a wound is a signal sent out to cells surrounding the wound bed for repair. And generally that starts with stasis, which is stopping of bleeding. That's then followed by inflammation and proliferation of the skin cells, usually the keratinocytes, and then remodeling and rebuilding of the architecture of the damaged tissue. And so this sequential phase of wound healing is something that's very important to be able to model to closely study this disease. So we took an attempt to model this in vitro by looking at specifically keratinocytes almost exclusively. We removed the immune system from this equation as well as the fibroblast and simply looked at keratinocytes at different stages of their growth. So let's talk a bit about the skin epidermis. So the skin epidermis is a very complex organ system. You actually have, I'm not sure if you're able to see my mouse here, my cursor, but you typically have a basal layer, which is the stem cell niche of the skin. That layer is characterized by the expression of a variety of proteins such as P63, a transcription factor, basal keratins, as well as integrins, which then, when these cells are ready to differentiate, usually upon sensing either damage or some kind of injury, you'll have differentiation upward towards the top of the skin. And this is also marked by a change in keratins from water-soluble keratins, such as keratin 14 and 5, to water-insoluble keratins for protection against permeable intruders. And so to model this wound healing, laboratories and investigators have largely used living systems, which makes sense because it's hard to study such a complex process outside of a living system. But the big challenge with in vivo models, which we also actually have in our laboratory, is the length of time it takes to build them and also the limitation in the high throughput capacity of these systems. And so we decided to explore another potential model. So what we've done here, we pretty much go to the clinic. Usually it's me that goes. We extract or we collect human foreskin tissue, neonatal foreskin tissue, and we bring that back to the lab. We separate the epidermis, which is what you see here, from the dermis. And the epidermis is, again, the portion that contains your keratinocyte population, which is key to complete the repair of the injury. We then cultivate these cells in keratinocyte stem cell media. And once we have a colony of cells that are grown, we perform spheroid formation assay. Now, the spheroid formation assay was developed to assess the capacity of epithelial stem cells to survive in suspension. Naturally, as I talked about earlier, keratinocytes adhere to a basement membrane by the use of integrins, rather. So once they're detached from that basement membrane, at least in the skin, you have something called suspension-induced cell death, or anoikis. This naturally causes cells, anoikis causes cells, or the process of anoikis causes cells to differentiate and die, but stem cells are usually capable of escaping this process and self-aggregate as multicellular spheroids. So by using this assay on our primary keratinocytes, we were able to assess for stem cell features in these cells. Now, before I go on, I want to note that investigators have looked at the potency, if you will, the stem cell features of keratinocytes before, but none have looked at it with high specificity in human skin, at least human keratinocytes. And that's largely because, obviously, it's quite challenging to get access to primary human tissue, and it's very difficult to work with it. But in essence, I'm going to run through this assay quickly. This is a tissue, skin tissue that we bring to the lab. We extract keratinocytes from this tissue, and then we grow these keratinocytes as 3D suspension cultures on agar beds. And this prevents cells from adhering to the bottom of the well. And then following up, we look at a variety of phenotypic changes and molecular changes in vitro. So one of the first things that we noticed was the vast majority of cells were not able to form these spheroids, which we expected. About 60% of cells, if you plate them in suspension, you have cell death taking place, anoikis. And if you try to take these cells that are dead, more or less, in suspension and place them back in culture, you find that they still don't grow. Nothing, they're not able to generate colonies. But a fraction of samples, some individuals had cells that were able to escape anoikis, about 41%, and actually form multicellular spheroids with this primary keratinocyte cell lines. And what you have here, if you place that spheroid back in culture, in 2D culture, you have, you can see here, small cells propagating from that spheroid. And now you can look at different gene expressions, such as involucrin, which is a differentiation marker. And what you find is cells from individuals that were able to form these spheroids have generally reduced levels of these differentiation markers relative to cells that were unable to survive that the spheroid formation assay. We see the same thing with EGFR, which is another marker of differentiation. And we decided to assess the ability of adult tissue to, and neonatal tissue to assess if there was a difference in the way they grew. And we found also that neonatal tissue was much better able to form spheroids than adult tissue. So this was quite intriguing. It was indicated that this is a stem cell, sort of a stem cell enrichment phenotype in neonatal tissue. So we decided to study this process more closely by transfecting our 2D cultured keratinocytes with a reporter plasmid expressing EGFP. And we then transferred the EGFP expressing cells in 3D culture. Now you have an EGFP expressing spheroid. And by using this tool, we can actually automate the localization of cells within that spheroids and sort of analyze how they move and migrate throughout the spheroid, how they conform themselves into spheroids, et cetera. And we can actually do time course assays using GFP as our reporter to assess what's happening to these spheroid forming cells when they are in suspension. Are they localized more at the center? Are they localized more towards the edge or the outside? And we find that over the course of 15 days, most cells generally moved towards the outside. You have a hollow center, some cells on the outside, and a cornified ring. And we knew it was cornified because when we stained for stem cell markers, keratin 14 and P63, we didn't find anything. And when we looked for involucrin mRNA expression, we found high levels of that gene expression within these rings, indicating that cells within these rings were cornifying and cells outside were able to escape it. So to closely study this, we then took cells from 3D and then placed them back in 2D. So we're no longer going from 2D to 3D, but now going from 3D back to 2D. And again, what you see here are microscopic cells migrating from the spheroid. You can actually maintain these cells in culture for about 30 days. You can see very nice cell populations continually proliferate, whereas mass cultured cells have a hard time doing so. If you look for stem cell markers, epithelial stem cell markers, you identify that they are present in the spheroid-derived population, but they are lacking at the same time point in the mass cultured population. Again, looking at EGFR, which is increasingly expressed as cells enter differentiation programs, we find that being present as we passage the mass cultured cells, but EGFR is relatively stable in the spheroid-derived population. The next thing we wanted to assess is what is the makeup, what is the population makeup of these spheroids? We know it's keratinocytes, but keratinocytes differentiate as we talked about earlier, and therefore they kind of take different lineages, if you will. And we decided to look at EGFR expression and integrin alpha-6 expression, which is, again, markers of differentiation and stemness, respectively. And we found that the spheroid-derived population had an enriched population of integrin alpha-6 high, EGFR low cells, which were able to remain in culture for over 40 days relative to the other fraction of cells. And if you stain that population of integrin alpha-6 high, EGFR low cells, we found that they express high levels of P63, and many cells also co-express both P63 and keratin 14, which is a key marker of the transient amplifying stem cell population of the skin. These cells were also able to regenerate colonies, while the opposite population of cells, EGFR high, integrin alpha-6 low, was unable to do so. We also find that subpopulation exclusively could regenerate spheroids in culture, but the opposite population was unable to. And so what we identified here was that these spheroids were able to form spheroids because they had this enriched, they were able to enrich for this population of epidermal stem cells. And so we became very intrigued by this process, understanding exactly what's taking place genetically at each stage of this assay. So we extracted RNA from the 2D culture, 3D culture, and again from 3D to 2D. And what we found was you can observe keratin 14 expression at each stage of the assay, but we see really nice expression of the basal keratins when cells are cultivated in suspension, 3D suspension culture, and also high expression when you transfer them to 2D. This is relative to the monolayer, to the autologous cultures. You also see, interestingly, that there's a high amount of differentiation taking place also in 3D. So this is consistent actually with what we saw earlier with that spheroid ring that remained there. And interestingly, when you take that spheroid and plate it back in 2D culture, you see a spike in genes involved in cell proliferation. So there's something being triggered by the spheroid attaching back into 2D culture to generate proliferation, which is a very similar response to injury, for instance, in the context of the skin. We also looked at self-renewal markers, and we also found that in 3D suspension culture, these cells had high expressions of self-renewal genes, indicating that there's a reprogramming that's taking place in 3D, and a completely different program is activated when the cells are plated back into 2D. But that program is distinct from the initial 2D program. And so to study that closely, we then took our EGFP expressing NHKCs, transferred them back into 2D culture, and then just study the propagating EGFP positive cells to understand exactly their growth dynamics. What we find is these cells retain high expression of the basal markers, keratin 14 and P63. They are able to regenerate whole colonies by themselves. Here is a single cell that we're able to track for 24 hours, and this is after about 15 days, and you can see it regenerating a nice colony. And we can also look at crystal violet blue staining of these attached spheroids and study the topography and the architecture of these cells as they propagate through the spheroids. And you can observe here a cornified-like ring with basal-like cells growing underneath, a feature that's strikingly similar to what you find in the natural architecture of the skin. And likewise, we looked at the average cell size, which is a feature also of stem cells. They tend to be small in size, the length of time in culture, and the number of population doublings that they were able to accumulate. And we found that the spheroid-derived cells outcompeted the mass-cultured cells from the same tissue. The next thing we wanted to look at was gene expression profile, transcriptomal profile. Before we looked at gene expression by PCR, this time we looked at entire transcriptomal expression. And what we found actually was 380 genes were upregulated when you took a 3D spheroid and played it back in 2D culture, and 185 genes were downregulated. And so we found that indeed this process is changing not just phenotypically, but also at a transcriptional level. And when you look at gene ontology programs that are activated in cells, you find things such as formation of skin, proliferation of epidermis, ectoderm commitment as the top programs being activated in these cells. And so I'm just gonna kind of speed up here. I believe I'm probably out of time. Let's see. Yes, so the reviewers actually wanted for us to assess a phenotypic function of these enriched stem cells of the skin. And so we decided to do organotypic cultures, 3D cultures. I'm not going to talk too much about this technique. It was established actually by our department at the Brigham years ago. But essentially the idea is you can recreate a synthetic skin in culture. And what you're looking for here are cells that grow above the white line here, which is your proliferative keratinocyte population. And so in a non-spheroid forming suspensions, they're not able to grow as you would expect. This is our negative control. In the monolayer from the non-spheroid forming group, again, very thin layers. Monolayer from the spheroid forming group, very thin layers, but then the spheroid derived population of cells, you can see very thick epidermal layers here. And that thickness can actually be quantified. What we found is these cells have enriched properties for stem cells, but also are better able to regenerate the entire skin, the skin epidermis. Okay, so in closing here, we believe we can actually use this assay as a high throughput system to study tissue repair of the epidermis. The first step is homeostatic maintenance, which is before any injury or disruption has taken place in cells. The next step is differentiation halt or reversal. This is after injury. This is us artificially inducing injury by plating cells in suspension. This triggers a flight or fight response, so to speak, where cells either form spheroids or die. The cells that form spheroids obviously are enriched for these stem cell reprogramming factors. When we take them back in culture, we believe this replicates the stress lineage activation that we see after injury of skin. And so these cells are primed to proliferate and regenerate the destroyed epidermis. That regeneration, that reconstruction is actually part of the repair and the restoration mechanism that we're hoping to study. And so by using the system, we can have a phase-by-phase system or model to investigate tissue repair ex vivo. And with that, I'm going to close, thanking my labs, my collaborators, and my funding sources. Thank you. I'll take questions.
Great, thank you very much. That was a great talk. Unfortunately, we don't have a lot of time for questions, but you do have some questions. If you will be able to answer that on the chat function, would be great. We just have to move over to the next speaker, which is the last speaker in this session. Let me just get your title right, sorry. So I'm introducing here to Mattias Pedicati from Politecnico di Torino, telling us about fibrotic cardiac tissue in vitro models based on bioartificial scaffolds. Mattia, when you're ready.
Yes, I will share my screen. Is it visible?
Yes, great.
Okay. Okay, hello to everyone. I briefly introduce myself. I am a biomedical engineering. I'm a postgraduate fellow at Politecnico di Torino, and I will start my PhD in November. And I will talk about our work on the design of an in vitro model of fibrotic cardiac tissue. Fibrotic condition occur in the cardiac tissue after myocardial infarction that cause massive loss and death of cardiomyocytes that are replaced by cardiac fibroblasts. In the infarcted area, cardiac fibroblasts proliferate and differentiate in their activated form, myofibroblasts. These myofibroblasts are able to produce stiffer and less functional extracellular matrix that is rich in collagen. At the end of this process, the aligned healthy cardiac tissue is replaced by randomly organized, less functional and stiffer fibrotic tissue. Myocardial infarction is a worldwide problem, is the main cause of mortality and morbidity worldwide. It causes more than 14 billion of deaths every year. And since heart cannot regenerate itself, the only available therapy right now is a heart transplantation. That unfortunately cause the onset of many comorbidities in patients and leads to many rehospitalization after 1 year of heart transplantation. And it, of course, cause a lot of high cost for the healthcare systems. So there is right now a urgent need for new therapies. In fact, many therapies are under investigation. But as we know, it's very difficult to move from the successful results of the lab test to the clinical trials. And to make the validation process safer and faster, we need a reliable platform for testing new therapies. Right now, the most used platform for testing is the in vivo testing of animals. But according to the 3R principle, the in vivo animal models has to be replaced, reduced, and refined, not just for an ethical concerning, but just also because they are poorly predictive about the human physiological response and can lead to many risks for later clinical trials. So in this scenario, in vitro models for testing new therapies can be a very useful tool, also to reproduce in a controlled way the complexity of human tissues. Currently, the state of art of in vitro models of pathological cardiac tissue is mainly based by hydrogels-based models that cannot provide adequate topographical and mechanical stimuli for cells. On the other hand, scaffold-based models are used mainly to reproduce healthy cardiac tissue through aligned substrates. But for both cases, mostly animal-derived cells are used. But since we know that we want to predict the human physiological response, of course, we need human cells, human-derived cells, or eventually single patient-derived cells, since also cardiac fibrosis has an high patient-to-patient variability in terms of extension and progression degree. So the aim of this work is to overcome the limits of the current state of art, producing an in vitro model of human cardiac fibrotic tissue. We choose polycaprolactone, PCL, to produce our scaffolds in order to give the adequate mechanical support to mimic the fibrotic condition and also to reproduce early stage fibrosis. We gave a random morphology to our scaffold. We used human cardiac fibroblast and we coated our scaffold with gelatin in order to sustain human cardiac fibroblast culture and extracellular matrix deposition. This project is part of the, this work is part of the BioRecAR project, a project that aimed to develop a new myocardial regeneration approach through an in situ direct reprogramming of human cardiac fibroblasts into functional cardiomyocytes. So an in vitro model is needed to test this new therapy, and this is the steps for the fabrication of an in vitro model. So at first, the scaffold design and production. We designed and produced a bidimensional scaffold through electrospinning technique to reproduce small thickness of infarcted tissue. And we use three-dimensional structure produced by melt extrusion additive manufacturing to reproduce great thickness of infarcted tissue. So we want to reproduce like physiological dimension of the infarcted area. So as I previously said, to give the right hint for the reproduction of early fibrotic heart tissue morphology, we gave random morphology to both scaffold typology. We use polycaprolactone with a low molecular weight as it is FDA approved, is biocompatible, it has a low melting temperature, and it's easy processable through many techniques, as it is also demonstrated by its wide use in literature. About the bidimensional scaffolds, we produced this bidimensional scaffold through the PCL electrospinning of random nanofibrous membranes. And we manage with the voltage and the collector distances to obtain nanometric and with less defect fibers. In the graphs, you can see the average fiber diameter and the average pore diameter. Three-dimensional scaffolds were produced by Rocket in vivo 3D printer. And by managing the printing parameters and customizing G-codes, we produce different self-supporting scaffold, as you can see in the pictures. Today, we will focus on the grid morphology scaffolds. We produced and designed two pore size scaffolds, one bigger and one small pore size, and both scaffold typology presents good interconnection rate. After this, next step was the surface modification. Since PCL is biocompatible but is not bioactive, we decide to coat it with the gelatin coating that And we decided to enhance the amount of gelatin grafted and the stability of gelatin coating with a polydopa adhesive pre-coating using the mechanism for muscle adhesion. It's known that dopa is an amino acid present in the foot protein of the muscle, and it can self-polymerize in a mild alkaline pH, creating an adhesive substrate that can bond with both inorganic and organic surfaces. So we basically immerse the scaffolds in a dopa solution and then in a gelatin solution for the gelatin grafting. Gelatin was chosen as an optimal substrate for the human cardiac fibroblast adhesion and proliferation. And since also is a collagen-derived protein, it was chosen also because it can mimic the fibrotic ACM composition. In addition, it's low cost and easy processable. After this, last steps was the biological validation of this model, and we cultured human cardiac fibroblast on polydopa and gelatin coated scaffold. And as a control, we also seeded and cultured cells on the PCL uncoated scaffold and on the polydopa coated scaffold. And as it demonstrates from the picture of the bidimensional and both also three-dimensional scaffolds with both the porosities, cells has a great viability and attachment on the polydopa gelatin coated scaffolds. And in addition, live dead staining after 24 hours reveals that no dead cells were spotted on all the scaffolds. After 7 days, polydopa gelatin scaffold coated scaffold results to be the best solution to enhance the proliferation and adhesion of human cardiac fibroblasts. In particular, in the bidimensional scaffolds, they completely covered and populated the surface of the scaffolds. In a three-dimensional scaffold with the bigger porosity, the pores were preserved and not populated, while in the three-dimensional scaffolds with the smaller porosity, all the porosity were closed and the scaffolds were completely populated. In addition, after 21 days, we evaluated the fibrotic SCM deposition by fibronectin and collagen for staining on polydopa gelatin coated scaffolds, and other fibrotic SCM protein staining as tenascin, laminin, and collagen 1 and 3 are ongoing. So summarizing in conclusion, cellularized bidimensional and three-dimensional bioartificial scaffolds were developed to reproduce cardiac fibrotic tissue with small and great thickness. Gelatin coating enhanced the cell attachment and viability, and scaffold composition, stiffness, and random architecture sustain cells culture up to 21 days and support the matrix deposition. In the future, so we want to conclude the fibrotic matrix proteins deposition and myofibroblast markers as alphasma staining after 21 days. And after all, we would like to test our advanced reprogramming therapy using bidimensional and three-dimensional model. This project is financed and supported by the European Research Council and as part of the Horizon 2020 program. And our partnership involves some Italian universities and research centers. And in addition, we are open to new collaborations. In conclusion, I would like to introduce you and to thank the BioRecart team, in particular, the principal investigator, Professor Valeria Chiono, and my colleagues, Geraldina Rocco, Irene Carmagnola, and Alice Zoso that participate and help me in this project. Also, I would love to invite you to follow us on our social medias and website, and also to submit your abstract and your work at our symposium at Termis World Congress 2020 entitled Advanced Therapies for Cardiac Regeneration. Thanks for your attention. I'm ready for your questions.
Thank you very much. That was a great talk. You have a few questions. Did you check the formation of ICM on the scaffolds through SEMAN studies?
Through what? I lost the connection.
Did you check the formation of ECM on the scaffolds through SEM studies?
Yes, we checked the. Yes, we checked the SEM by the staining of these just two proteins, but it lacks from the other proteins, and we just stained the protein, the matrix that was deposited directly on the scaffolds with the immunostaining.
Cool. Another question, did fibroblasts migrate inside the PCL scaffold? And were there any histological or microscopical cross-sectional images made to prove that?
We are about to evaluate that. We are about, in fact, to evaluate the proliferation rate inside the, of course, mainly the three-dimensional scaffolds. We will have also to evaluate to cut the scaffolds to see if the proliferation goes inside. It looks from the images that they proliferate completely inside, but we need more proofs. So we know that with a smaller porosity, they completely enclose the pores, but we don't know any certain results about the inside part.
Cool. Another one, did you alter the stiffness to match healthy and fibrotic tissues?
Stiffness helped the fibrotic tissue to, the cardiac fibroblasts to differentiate, actually. We didn't measure, but okay, we know that it helps cardiac fibroblasts to differentiate in their fibrotic phenotype.
Mm-hmm. And I was just wondering, when you talked about human cardiac fibroblasts, I was just wondering if these were patient-derived or they were iPS-derived cells, or what is that lines?
They are patient-derived cells. They were taken from the left ventricle, I think, but yes, it's a patient-derived. They are healthy human cardiac fibroblasts, and they are, yes, patient-derived.
Cool, thank you.
We are also currently evaluating the behavior of some pathological cardiac fibroblasts, but everything is ongoing now.
Mm-hmm. You do have a few more questions, but I'm afraid we are short in time, so we should close the session. If you're happy to answer the questions over the chat, that would be great. So with this, we would like to thank all the speakers from session 2. That was a great session, and I believe you have a break now, so I'll see you soon after the break. Thank you very much.